Regardless of whether the original form of energy is sunlight or food, it must be converted to the cellular energy of ATP.
Electron transport requires a membrane in order to work. In prokaryote cells, those bacteria and bacteria transport takes place in the cell's plasma membrane in folded areas called membranes.
For most organisms, this conversion is accomplished through cellular respiration, pathways in which glucose is broken down and the energy extracted is converted to ATP.
The pathways of cellular respiration include 1. Glycolysis, 2. Conversion of acetyl-CoA, 3. Krebs cycle, and 4. Electron transport. Electron transport is the most complex and productive pathway of cellular respiration. During the aerobic respiration, the ETC produces 34 of the 38 ATP molecules obtained from every molecule of glucose.
Whichever type of membrane houses the cell's electron transport chains, plasma membrane, inner membrane of mitochondria, or the thioclone membrane of chloroplasts, cells have many ETCs running continuously to produce the ATP energy required for cells to survive and thrive.
Most of the ATP made in cellular respiration comes from the release of energy of a series of oxidation-reduction reactions between molecules embedded in the plasma membrane, prokaryotes or mitochondria eukaryotes. Electron transport is divided in 3 main events.
In the electron transport chain, these electron carriers are oxidized, transferring their electrons to the carrier molecules embedded in the ETC membrane.
In aerobic respiration, these electrons are passed from one carrier molecule to another in a series of oxidation-reduction reactions, and ultimately to the final electron acceptor, oxygen, that combines with hydrogen, resulting in a water H2O oxidation-reduction reactions.
During glycolysis, synthesis of acetyl-CoA and Krebs cycle, the electron carriers NAD+ and FADH are reduced to form NADH and FADH2. Respectively, these molecules are similar to rechargeable batteries and where NAD+ and FADH are reduced. This means that they accept and carry electrons and hydrogen ions, potential energy that can be used later in cellular respiration.
The energy from each electron being passed down the chain is used to pump a proton H+ hydrogen each carrier molecule from one side of the membrane to the other. This creates a proton gradient, a type of concentration gradient, difference in concentration of a substance between two sides of membrane, and gradients are potential energy available for cellular work.
The hydrogen ions on the side of the membrane where most concentrated will eventually flow back across the membrane, down the proton gradient, through an enzyme called ATP synthetase.
As each H moves back across the membrane, the enzyme ATP synthase phosphorylates adds a phosphate to adenosine diphosphate, ADP, to make the high-energy molecule ATP, which can be used for many different energy-requiring reactions throughout the cell. All organisms use cellular respiration. Cellular respiration is important because all cells use oxygen to convert food into energy. Cells would die without cellular respiration because they would not have the oxygen needed for food conservation. The cells would also have issues getting rid of their waste products.
Cellular respiration is the process by which energy is released from the breakdown of glucose in the presence of oxygen. The formula for cellular respiration is oxygen plus sugar converts to carbon dioxide plus hydrogen plus energy.
This means that air is important to us because if we don't breathe, we die. Cellular respiration occurs in the cytoplasm and mitochondria. Cellular respiration consists of a sequence of steps, which can be divided into 3 main stages: glycolysis, Krebs cycle, and electron transport chain, or ETC. Glycolysis occurs in the cytoplasmic fluid of the cell. Oxygen is not needed for this phase, and it is called anaerobic, which means without air. Results of glycolysis: 2 ATP is used. 4 ATP is made. The net gain of 2 ATP, 2 NADPH is made. Two pyruvic acids is then used in the next cycle.
Glycolysis is the anaerobic process, which means it does not require oxygen. It takes place in the cell cytoplasm. It requires ATP molecules. ATP is the fuel used by all human cells for cellular respiration. The net end result of glycolysis is 2 molecules of ATP, 2 molecules of pyruvic acid, 2 molecules of water, and 2 molecules of NADH.
Oxidation is changing the way molecules share electrons. NAD+ is a coenzyme, a helper, whose role is to transport electrons. NAD+ is an oxidation agent. When NAD+ picks up electrons for transport, it becomes NADPH+. When a phosphate group is added to an ADP molecule, the ADP molecule is transferred into an ATP molecule.
Glycolysis is the process of breaking down glucose. It's the most simple form. Glucose can be thought of as a 6-carbon molecule. The result is a 6-carbon sugar molecule that is called fructose. It is important to notice that the phosphates have been added to the molecular equation. These phosphates will be shared as enzymes continue to act on our simple glucose molecule. This process uses 2 ATP molecules.
Sugar oxidation and ATP formation, now the NAD+ comes into play. The NAD+ comes along and rearranges the way our 3 carbon sugar molecules are sharing electrons. The NAD+ becomes NADPH+, an electron donor. More enzymes come along and perform their magic. More phosphate molecules are shuffled around. The result of sugar oxidation process is 2 NAD+, 2 H2O, 2 molecules of pyruvic acid, and 4 ATP molecules.
The end result is 4 ATP molecules, 2 H2Os, 2 pyruvic acids, and 2 NADH molecules. 4 ATP molecules minus 2 ATP molecules equals 2 ATP molecules net the 2 pyruvic molecules are either passed along and used in the Krebs cycle to create more ATP.
Krebs cycle is an aerobic process, which means oxygen is required. The Krebs cycle occurs in the mitochondria of the cell, the cell's power plant. Each glucose molecule creates 2 pyruvic acids. The Krebs cycle breaks down 1 pyruvic acid at a time. It begins at the end with an oxalic acid at a 4-carbon molecule.
The Krebs cycle is an 8-step cycle. Each pyruvic acid molecule yields 3 carbon dioxide, 1 ATP, and 5 reduced enzymes. The CO2 are eventually released via respiratory expiration. The NADH and FADH2 molecules are high-energy electron donors that will carry their electrons in their electron transport chain.
Prior to entering the Krebs cycle, pyruvate must be converted to an ACETYLCoA. This is achieved by removing a CO2 molecule from pyruvate and then removing an electron to reduce an NAD+ into NADPH. An enzyme called coenzyme A is combined with the remaining acetyl to make acetyl-CoA, which is then fed into the Krebs cycle. Citrate is formed when the acetyl group from acetyl-CoA combines with oxalate from previous Krebs cycle. Citrate is converted into its isomer isocitrate.
Isocitrate is oxidized to form the 5-carbon A-ketoglutarate. This step releases 1 molecule of carbon dioxide and reduces NAD+ to NADH2+. Malate is oxidized to oxalate, reducing NAD+ to NADH2+.
We are now back at the beginning of the Krebs cycle. Because glycolysis produces 2 pyruvic molecules from 1 glucose, each glucose is processes through the Krebs cycle twice. For each molecule of glucose, 6 NADH and 2 FADH and 2 ATP. Krebs cycle oxidation changes the way molecules share electrons. NAD+ is a coenzyme who transport electrons. NAD+ oxidizes. When NAD+ picks up electrons for transport, it becomes NADH. FAD and FADH2 is non-protein. chemical compound that binds to the phosphate group of the ADP molecule. NAD plus and NADH helps rearrange electrons in a chemical reactions. FADH2 has significantly higher energy than FAD. The Krebs cycle, pyruvic acid releases carbon dioxide. The CO2 is eventually released back into the atmosphere by respiratory expiration. NAD plus picks up some hydrogen and acetic acid is formed. Acetic acid is bound to coenzyme A and acetyl CoA is formed. Acetyl CoA enters the Krebs cycle. Acetyl CoA's role in Krebs cycle is to transport acetic acid from one enzyme to another.
The Krebs cycle uses the products of glycolysis to produce two ATP molecules for each molecule of glucose. The NADH and FADH2 that are high-energy coenzymes are byproducts of the Krebs cycle and continue on to fuel the electron transport chain.